welcome to the Deep Dive. Today we're diving into the world of feline neuroimaging. Get ready to explore a tool that could revolutionize how researchers study cat brains. We've got this super interesting research article titled Automated Registration-Based Skull Stripping Procedure for Feline Neuroimaging. Now, I know what you're thinking, skull stripping. Sounds kind of intense, right? And yeah, it is. But it's a crucial process in neuroimaging, and this new tool, CARS, is making waves. To help us unpack all of this, we have an expert in veterinary neuroimaging joining us today. Welcome to the show.
It's great to be here. Thanks for having me.
Before we get into the nitty-gritty of this new tool, can you walk us through why skull stripping is so important in neuroimaging? It's not like we're dealing with actual scalpels here, right?
Right. No scalpels involved. It's all digital. To analyze brain scans effectively, we need to isolate the brain from all the surrounding tissues, and the skull is a big one. Imagine trying to examine a delicate flower encased in amber. The amber might be protecting it, but it also obscures the finer details. Skull stripping is like digitally removing that amber, allowing us to get a clear, unobstructed view of the brain itself.
That makes perfect sense. So it's all about getting the clearest possible picture of the brain for analysis. But I imagine accurately distinguishing between brain tissue and bone in a scan can get tricky, especially with all the folds and fissures of the brain.
You're absolutely right. The brain is incredibly complex. Accurately segmenting it from the skull in scans requires a high level of precision. One wrong move and you could end up accidentally editing out part of the hippocampus, or mistaking a blood vessel for a fissure.
Yikes. Talk about a high-stakes game of digital surgery. So traditionally, how has the skull stripping been done? Has it always been a meticulous manual process?
For a long time, it was primarily a manual process. Researchers would painstakingly trace the outline of the brain on countless MRI slices, which, as you can imagine, was incredibly time-consuming and prone to human error.
I can only imagine the eye strain. Sounds like a recipe for caffeine overload. So this new automated tool, CARS, is here to save the day, or at least save researchers a whole lot of time and headaches.
You got it. And it's not just about saving time, though. That's certainly a bonus. Automated tools like CARS have the potential to significantly increase the accuracy and consistency of skull stripping. And that leads to more reliable research results.
Okay, so we've established that skull stripping is crucial for analyzing brain scans, and that CARS is stepping in to automate this process. But how does it actually work? What's the magic behind this tool?
CARS, which stands for Cat Automated Registration-Based Skull Stripper, utilizes a really innovative approach. It involves a pre-made template of a cat brain. This template, along with the MRI images from the subject, is fed into a powerful software program called ANTs.
ANTs, as in the insects.
You could say that, though in this case, ANTs stands for Advanced Normalization Tools. It plays a key role in how CARS works. It takes that template brain and essentially warps and molds it to match the unique shape of the subject's brain within their MRI scan.
Wow, so it's like having a digital sculptor on hand, meticulously shaping that template brain to perfectly fit the contours of each individual scan.
That's a great way to put it. And the software uses sophisticated algorithms to do this, ensuring a high degree of accuracy and precision.
That's incredible. But how do we know CARS is actually doing a good job? I imagine there are ways to test its accuracy compared to the traditional manual methods.
You're right. Simply trusting a new tool without rigorously testing it wouldn't be very scientific, would it? That's where things get really interesting. That's where this particular research article comes in. The researchers wanted to see how CARS stacked up against the gold standard of manual segmentation and even a few other automated methods. And to do that, they used some pretty sophisticated metrics to evaluate the accuracy of each approach.
Like those brain-bending terms I came across in the article, dice similarity coefficient and Hausdorff distance. I have to admit, those made my head spin a little.
They can sound intimidating, but trust me, they're not as complicated as they seem. Think of the dice similarity coefficient, or DSC, like this. Imagine you have two outlines of a cat brain, one created manually and one generated by CARS. The DSC measures how much those two outlines overlap.
So a higher DSC score means a better match between the automated and manual outline.
Exactly. A perfect score of 1 would mean CARS recreated the manual segmentation flawlessly. And the researchers found that CARS consistently achieved very high DSC scores.
Okay, so far so good.
Okay.
What about Hausdorff distance? That one sounds a bit more intimidating.
It's really just a way to measure the biggest difference between the two outlines, the automated and manual ones. Imagine you're measuring the distance between two coastlines. Hausdorff distance tells you the furthest point on one coastline from the other.
So a smaller Hausdorff distance means the automated outline is more faithful to the manual one.
You've got it. And again, the researchers found that cars performed exceptionally well on this metric too. They even looked at the 99th percentile of the Hausdorff distance, which basically helps to smooth out any tiny, insignificant variations.
So they're really putting cars through the ringer, making sure it's accurate and consistent. But tell me about the cats involved in this study. Were they all just your average house cats?
Actually, they used MRI scans from a diverse group of 82 cats, a mix of both hearing and deaf cats, ranging in age from 6 months to over 6 years old.
That's a pretty good sample size. And I'm curious about the inclusion of both hearing and deaf cats. Was there a specific reason for that?
That's a great question. You see, the researchers wanted to make sure cars could handle variations in brain structure. By including deaf cats, they could see if cars was thrown off by any subtle differences in brain anatomy that can occur with hearing loss.
That makes a lot of sense. It's like giving cars a real-world test drive to see how it handles different types of roads, or in this case, different types of feline brains.
Exactly. And remember, this study wasn't just about proving that cars could strip skulls. It was about thoroughly validating its accuracy and consistency compared to established methods, and seeing if it could live up to its full potential.
Rigor is key. Okay, so tell me, how did cars actually perform? Did it live up to the hype?
Well, the results were pretty impressive. When they compared cars to the gold standard of manual segmentations, the median dice similarity coefficient score was a remarkable 0.987.
Wow, that's incredibly high. That means the overlap between the automated outlines generated by cars and the manually created ones was almost perfect.
Exactly. It blew the other automated tools out of the water. And here's where it gets even more interesting. They ran cars multiple times on the same images to see how consistent the results were. And get this. The dice similarity coefficient score for those between-run comparisons was a near-perfect 0.998.
That's remarkable. It means cars was incredibly reliable, producing nearly identical results each time like a well-oiled machine.
Precisely. That level of consistency is crucial for researchers. Now, remember how we talked about the Hausdorff distance, that measure of the biggest discrepancy between the outlines? Well, for cars, the median score was only 0.866 millimeters.
So less than a millimeter difference. Considering we're talking about the intricate structures of a cat brain, that's incredibly precise.
It is. And even when they compared the Hausdorff distance scores between those multiple cars runs, it was even smaller, essentially the absolute smallest difference their instruments could measure. Talk about precision.
Now, that's what I call accuracy. But I have a feeling even cars isn't entirely foolproof. Were there any situations where it struggled?
You're right, no tool is perfect. Even with its advanced algorithms, there were a handful of cases where the automated masks needed a little bit of tweaking.
Every superhero has their kryptonite, right? So what were cars' weaknesses?
Interestingly, the areas that seemed to throw cars off were primarily concentrated around the dorsal cerebellum and the front and back edges of the cortex.
Those are pretty specific areas. Any ideas why those regions might have been particularly tricky for cars to handle?
It seems like the presence of sinuses within the skull in those areas, along with a structure called the bony tentorium in a cat skull, might have created some challenges for the algorithms to perfectly differentiate between bone and brain tissue.
Interesting. So even with all its sophistication, cars can still fall prey to the intricacies of feline anatomy.
It highlights just how complex skull stripping can be, even for advanced technology. But the good news is, even in those cases, the corrections needed were relatively minor.
So cars still saves researchers a ton of time, even with those minor adjustments.
Absolutely. Out of the 60 scans they used for this part of the validation, only 5 needed any manual corrections at all. And those corrections took significantly less time than it would have taken to manually segment the entire brain from scratch. We're talking about a difference of hours versus minutes.
That's a game changer for research efficiency. It sounds like cars is revolutionizing how brain imaging studies can be done, opening up possibilities for larger studies and faster results. But I imagine the researchers didn't just stop at comparing cars to manual segmentation. Did they pit it against any other automated tools to see how it measured up?
You're exactly right. They wanted to see how cars compared to other available tools. They included a tool called BET, which was specifically designed for human brains, to see how well it would fare with feline scans. And they also included Atlas Brex and ANT's Ants Brain Extraction.esh, which are a bit more flexible and can be adapted for different species.
So a real test of species-specific versus more general-purpose tools. What were the results of this automated skull stripping showdown?
Well, as we've discussed, cars emerged as the clear champion in terms of both accuracy and consistency. It significantly outperformed the other automated tools, including BT, which, as you might expect, struggled with those feline brains.
Poor Bet, thrown into the ring without a scratching post. It really highlights the importance of having tools specifically designed for the unique anatomy of different species.
Absolutely. Now, while cars excelled in its performance, it's important to remember that no tool is without its limitations.
Okay, let's unpack that a bit. Every superhero has their kryptonite. What are some of the potential limitations of cars that researchers should be aware of?
That's right. Even the most advanced tools have their limitations. One thing to remember is that cars' accuracy is dependent on the quality of the MRI scans themselves. If you feed it a blurry, low-resolution scan, you can't expect it to magically produce a perfect brain segmentation.
Garbage in, garbage out, as they say. So cars can't work miracles on bad data.
Exactly. And while those instances were rare in the study, there might be cases where manual adjustments to the car's generated masks are still necessary, especially in those tricky areas we talked about earlier, like around the sinuses and the bony tentorium.
Right, those anatomical nuances that even cars find challenging. It's a good reminder that even with automation, human expertise is still essential.
Absolutely. It's about using technology to enhance our abilities, not replace them entirely.
It's a partnership, really. Humans and AI working together to achieve the best possible outcome. So with these limitations in mind, do you think there are any specific areas where further research and development of cars could be beneficial?
That's a great question. One area that comes to mind is exploring how cars performs on cats with significant brain abnormalities, such as tumors or lesions.
That makes sense. The study focused on a relatively healthy group of cats, both hearing and deaf. But what happens when you throw a brain tumor into the mix? Could that potentially confuse the algorithms?
It's definitely a possibility. Large tumors or lesions could alter the shape of the brain enough to potentially impact cars' accuracy. Further research in this area would be valuable to see if the algorithms need any tweaking to handle these types of cases.
So testing cars on a more diverse range of clinical cases could help to refine the tool even further and expand its applicability. Are there any other areas where you see potential for improvement?
Another avenue worth exploring is how cards could be adapted for use in other imaging modalities, such as CT scans. Right now, it's specifically designed for MRI, which is a powerful tool, but CT is more widely available in veterinary practices.
That's a great point. Having a CARS-like tool for CT scans could be a game changer for veterinary neurology, making advanced brain imaging analysis more accessible to a wider range of clinicians.
Precisely. It could democratize access to these powerful tools and potentially lead to earlier and more accurate diagnoses for a wider range of neurological conditions in cats.
I'm really starting to grasp the potential impact of cars here. It's not just about stripping skulls. It's about opening up a whole new world of possibilities for feline neuroimaging. But before we get too far ahead of ourselves, let's rewind a bit and talk about how this whole validation process actually worked. The article mentioned that they used a multi-step approach to really put cars through its paces. Can you walk us through that?
Of course. They were very thorough in their approach to validating cars. Remember those 82 MRI scans we discussed earlier?
Yeah.
Well, they used 60 of those scans for this specific part of the validation process.
60 scans. That's a lot of brain data. So what do they do with these scans? Do they just let cars loose on them and see what happened?
Not quite. Remember, they needed a gold standard to compare cars' performance against. So they had an expert meticulously hand segment the brains in all 60 of those MRI scans. Think of it like having a master chef bake a perfect cake that all the other cakes will be judged against.
I like that analogy. So this expert segmented data set became the benchmark for evaluating how well cars could replicate human-level accuracy.
Exactly. And to add another layer of rigor, they also had 2 additional independent raters, each manually segment a subset of 8 of those scans. This helped them establish how consistent manual segmentation is in general.
Makes sense. Even experts can have slightly different interpretations, right? It's important to account for that human variability factor.
Precisely. It helps to create a baseline for comparison. So with the gold standard segmentations and the baseline for human variability established, they were ready to let cars loose on those 60 scans.
It's like the ultimate bake-off. Cars versus the human experts. So what happened? Did cars rise to the occasion?
It absolutely did. As we've discussed, it achieved those impressive dissimilarity coefficient and Hausdorff distance scores, consistently producing highly accurate segmentations that were nearly indistinguishable from the manually created ones.
And like cars took home the gold medal in this bake-off. But you mentioned they didn't stop there. They also ran cars multiple times on the entire data set at 82 scans, right?
You're right. They wanted to go beyond just a single comparison and really assess Cars's consistency and reliability. They ran it multiple times on all 82 scans and then compared the results of those runs to see if there were any significant variations.
So it's like checking if Cars was giving the same answer to the same problem every single time.
Exactly. Consistency is key, especially when you're talking about a tool that could be used in research and potentially even in clinical settings. And the results were reassuring. Cars demonstrated remarkable consistency, producing nearly identical segmentations each time it was run.
That's reassuring to hear. But I'm also curious about those other automated skull stripping tools you mentioned earlier. BET, Atlas Brex, and A&T's Ants Brain Extraction.SHA. What was the purpose of including them in the study?
The researchers wanted to see how Cars stacked up against other available tools, both those specifically designed for brains and those with more flexibility. Remember, BET was developed for human brains, while Atlas Brex and A&T's Ants Brain Extraction.SH have a broader scope.
So it was a real head-to-head, or should I say brain-to-brain, competition to see which tool reigned supreme.
Exactly. They ran each of these tools on that same set of 60 scans, comparing their performance to the gold standard manual segmentations using their same rigorous metrics: Dice similarity coefficient and Hausdorff distance.
Okay, I'm on the edge of my seat. Give us the rundown of this epic skull-stripping showdown. Who came out on top?
Well, as we've discussed, Cars emerged as the clear frontrunner, significantly outperforming all the other tools in terms of both accuracy and consistency.
Sounds like Cars was in a league of its own. But what about the other contenders? Did any of them put up a good fight?
While none of them could quite match Cars's stellar performance, there were some interesting observations. Atlas Brex, for example, showed some promise. It delivered good results on about half of the images.
So a 50-50 chance of success. Not too bad, but I imagine consistency was an issue.
Exactly. It was a bit of a mixed bag with Atlas Brex. Some of the masks were great, while others had noticeable errors and misalignments.
So not quite ready for prime time, at least not without some further refinements. What about A&T's Ants Brain Extraction.SH? How did it perform?
A&T's Ants Brain Extraction.SH was actually the most consistent of the bunch, producing decent masks for almost all the images.
So a solid effort, but still not quite as accurate as Cars.
Right. It seemed to struggle a bit more with those fine details, those tiny discrepancies that can make a big difference in neuroimaging analysis. And of course, poor BET, our human brain specialist, didn't fare so well in the feline arena.
Yeah, those feline brains are a different breed altogether. But it really highlights the importance of using the right tool for the job. And in this case, Cars has clearly emerged as the superior tool for feline neuroimaging.
I'd have to agree with you there. And, this whole validation process, as rigorous as it was, is really just the beginning for Cars.
What do you mean? It sounds like it passed with flying colors.
It did. But think of it like this. This study was essentially Cars's debut performance. Now it's up to the research community to take this tool and really put it through its paces in a variety of research settings.
So this is really just the tip of the iceberg when it comes to Cars and its potential applications.
Exactly. And this is where things get really exciting, because the implications of this technology extend far beyond just automating a tedious task.
I'm intrigued. What kind of impact do you think Cars could have on the field of feline neuroimaging as a whole?
Well, imagine a world where we can study feline neurological disorders with the same level of detail and precision that we currently reserve for human patients.
That's a pretty amazing thought. We could finally start to unravel some of the mysteries surrounding those complex feline brains.
Exactly. Cars opens up the possibility of studying brain conditions in cats on a much larger scale. And remember how we talked about those big studies being crucial for reliable results? That's where this technology can make a real difference.
It's like finally having a telescope powerful enough to see those distant galaxies we could only dream of before. What kind of neurological conditions are we talking about here? What could Cars help us understand better?
Oh, the possibilities are vast. Take feline cognitive dysfunction, for example, a form of dementia that affects senior cats. With Cars, we could potentially identify those subtle brain changes associated with this condition much earlier, paving the way for earlier interventions and maybe even new treatments.
Wow, that would be huge. We always talk about early detection being key. And it sounds like Cars could be a powerful tool in making that a reality for our feline patients.
Absolutely. And it's not just about diagnosis. This technology could revolutionize how we evaluate the effectiveness of new treatments. Imagine being able to track the progression of a neurological condition in a cat over time, monitoring how their brain responds to a particular medication or therapy with a level of detail we could only dream of before.
We'd be moving beyond those anecdotal, it seems to be helping observations, to a more data-driven approach to veterinary neurology. That's incredible.
It really is. And because cars can significantly speed up the research process, it has the potential to accelerate the pace of discovery. Scientists can devote less time to the tedious task of manual segmentation and more time to analyzing data, testing hypotheses, and collaborating with other researchers.
It's like cars is breaking down those barriers to scientific progress, fostering a more interconnected and dynamic research community.
Precisely. And with more collaboration comes faster innovation, and ultimately a deeper understanding of the feline brain and the conditions that affect it. This translates into better care for our feline companions.
It's mind-blowing how a tool initially designed for a very specific task, like skull stripping, can have such far-reaching ripple effects across an entire field of study. It's incredibly inspiring.
It really is. But while we're riding this wave of enthusiasm, it's also important to acknowledge that CARS is not a magic solution. It's a powerful tool, yes, but it has its limitations.
Right, we talked about how important those high-quality MRI scans are for accurate results, and that even cars can stumble over those tricky anatomical areas like the sinuses.
Exactly. It's crucial for researchers to be mindful of these limitations and not view CARS as a set-it-and-forget-it solution. Human expertise and oversight are still paramount.
It's a partnership, as we discussed. Technology working hand in hand with human knowledge and experience to achieve the best possible outcome for our patients.
I couldn't agree more. And as technology continues to evolve, I have no doubt we'll see even more sophisticated tools emerging, further pushing the boundaries of what's possible in veterinary neuroimaging.
It's an exciting time to be in veterinary neurology. A huge thank you to you for joining us today and sharing your expertise. It's been an incredibly enlightening deep dive.
The pleasure was all mine. Thanks for having me.
And to our listeners, we hope you found this deep dive as fascinating as we did. Be sure to check out the show notes for links to the research article we discussed and other resources if you'd like to explore this topic further. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.